Welcome to the Deep Dive. Today, we're going deep on a case report about a mixed breed dog.
Oh, very cool.
Yeah, it was published in Frontiers in Veterinary Science.
Okay.
And it's about a newly discovered genetic mutation.
Interesting.
It centers around hereditary myotonia,
Okay.
which affects muscle relaxation.
So the muscles stay contracted for too long.
For longer than usual.
Right.
Exactly.
Okay.
So what makes this case so unique? I mean, we've seen hereditary myotonia before.
Right.
But this is about a mixed breed dog.
Yeah, so we've seen hereditary myotonia in purebred dogs before.
Yeah.
But this report focuses on a mixed breed dog.
Okay.
And it has a never-before-documented mutation.
A brand new mutation.
Yeah, complex mutation of the CLCN1 gene.
The CLCN1 gene.
Yeah.
All right, so let's get into the details.
Sure.
Tell me about this dog.
So it was a 4-month-old male mixed breed dog.
Okay.
And he was presented for a stiff gait after being startled. Yeah, and this symptom had been present since puppyhood.
So like from when he was really young.
Yeah.
A puppy.
Yeah, exactly.
Wow.
Interestingly, the dog also exhibited generalized muscle hypertrophy.
So his muscles were unusually large.
Precisely.
Okay.
And the veterinarians were able to easily induce myotonia.
Okay.
Particularly when the dog was asked to jump.
Wow. I can imagine that was quite a sight.
Yeah.
Watching this dog.
Yeah, it's pretty dramatic to see.
Yeah.
The report also mentions upper lip myotonia.
Oh, interesting.
Yeah, that was observed during handling.
Okay.
Due to the dog's defensive behavior.
So like when they were examining him and he was kind of nervous?
Yeah, so this myotonia is thought to be triggered by stress.
Okay.
You know, in those situations.
Yeah, that makes sense.
Yeah.
So despite these episodes of muscle stiffness,
right,
was the dog in any pain?
Thankfully, no.
Oh.
The owners reported no pain and really no significant impact on his quality of life at all.
Oh, that's good.
Yeah, and this is a crucial detail. Yeah.
Because it speaks to the really variable nature of hereditary myotonia.
Okay.
Some dogs can be severely affected.
Right.
But others, like this one, experience minimal impact.
Yeah, I guess that's good for him.
Yeah.
At least in this case.
Yeah.
So how did the vets confirm that it was hereditary myotonia?
So they conducted both electromyography, or EMG, and genetic testing.
Right. So EMG, what did that show?
So the EMG showed myotonic potentials in various muscles.
Myotonic potentials.
Yeah, which are essentially bursts of electrical activity that are a hallmark of myotonia.
So that's how they could tell something was up with his muscles.
Right. Yeah, they're like little signatures of myotonia.
Okay, so that's the EMG.
Yep.
What about the genetic testing? How did they do that?
So they took blood samples from the dog, his mother, 4 littermates.
Oh, wow.
And then an unrelated wild-type dog for comparison.
Okay, so they were really looking at the whole family.
The whole family.
To see, this family-wide approach let them analyze the inheritance patterns.
Very cool. All right, and what did they find?
So they discovered a complex of mutations.
Okay.
In Exon 15 of the CLCN1 gene.
That same gene again?
Yeah, and this gene is responsible for coding the chloride channel.
Okay.
That's crucial for muscle relaxation.
So the mutations are messing with that chloride channel?
Exactly. Yeah, they disrupt the normal function of the chloride channel.
Okay.
Yeah.
So they lead to a premature stop codon.
Right.
Which results in the
truncated protein.
Truncated protein.
Yeah, and so it's basically dysfunctional. Okay,
so the recipe for the chloride channel is like incomplete.
Yeah, that's a good way to put it.
And that's causing the myotonia.
Right.
Okay.
The genetic investigation of the family revealed that the mother and 3 littermates were carriers of the mutation.
So they had 1 copy of the bad gene and 1 copy of the good gene.
Exactly. And then 1 littermate was wild type, meaning they had 2 normal copies of the gene.
Okay, so this points to autosomal recessive.
Exactly.
Meaning you need 2 copies of the bad gene.
You need 2 copies to express the condition.
To express the condition.
Right.
Okay, and that's what they saw. Right. Because only the one dog...
Only the dog with 2 copies.
With 2 copies had the myotonia.
Had the myotonia, exactly.
Okay, so now we have a diagnosis.
Right.
How did they treat him?
So, interestingly, because the dog wasn't severely affected, they didn't actually start any treatment.
Oh, wow.
Yeah, but they really emphasized owner education.
Okay.
About the condition and its hereditary nature.
To avoid breeding.
To prevent further breeding,
Of dogs that might have this.
Exactly.
Okay, that makes sense.
Yeah.
Responsible breeding.
Yeah.
So what happened to the dog after the diagnosis? Did his condition get worse?
So fortunately, no.
Right.
The report includes a 2-year follow-up.
Oh, wow.
And the dog was doing well. He exhibited normal growth and behavior and no worsening of his myotonia.
Okay.
Interestingly, the muscle hypertrophy persisted.
Oh, so his muscles were still big.
Yeah, but it didn't seem to bother him or affect his well-being.
So this particular mutation, it causes the myotonia.
Yeah.
But it doesn't seem to...
It doesn't seem to be progressive.
It was like a really bad debilitating disease.
Right. It seems to be a mild form.
Okay.
At least in this case.
That's good.
Yeah.
So we've covered like the dog's journey.
Right.
And how they diagnosed him.
Yeah.
But like, what's the significance of this case?
Sure.
Like, why is it important?
Well, I think this case is significant for a couple of reasons.
Okay.
First, it confirms that hereditary myotonia can occur in mixed breed dogs.
Okay.
And specifically hereditary myotonia linked to the CLCN1 gene.
Okay.
You know, for a long time, research really focused on purebred dogs.
Yeah.
So this finding really broadens our understanding of how common this condition is.
Okay, so it's not just a purebred dog problem.
Exactly.
Interesting. Okay. What else makes this case stand out?
Well, the identification of this brand new mutation in the CLCN1 gene, it's really groundbreaking. It adds to our knowledge of, you know, all the genetic variations that can cause hereditary myotonia.
So that's like the crux of the matter.
Yeah.
Right? This discovery could change how we understand that gene.
Absolutely.
And how it affects muscle function.
Yeah, and the report goes into a lot of detail.
Oh, really?
Yeah, about the complex mutation.
Okay.
Even illustrating how it leads to that truncated protein.
I'm looking at that figure now.
Yeah.
It's really amazing how they pinpointed exactly where that mutation is in the gene.
Yeah, it is remarkable.
So what is it exactly?
So they found a complex of mutations.
Okay.
A deletion followed by an insertion. And it's denoted as C.16361639, Dallancy ACGGG.
Okay.
And then there's also a substitution denoted as C.1644AT.
Okay, and all of that is an exon 15.
Precisely exon 15 of the CLCN1 gene.
Of that gene.
Yeah.
Okay.
And that's a crucial part of the gene that provides the instructions for building that functional chloride channel.
Right. And this mutation messes that up.
Yeah, it disrupts the whole process.
So how does that lead to the symptoms of myotonia?
Well, the mutation creates what's called a premature stop codon.
Okay.
Which basically tells the cell to stop reading the genetic instructions too early.
Oh, so it's like an incomplete recipe.
Exactly. And that results in a truncated protein.
Yeah, we talked about that.
Yeah, a protein that's missing a really important part.
So it can't do its job.
Right. Instead of a fully formed and functional chloride channel, you get this incomplete version that can't do its job properly.
So the muscles can't relax.
That's exactly right.
Because the chloride channel isn't working.
Right.
Okay.
And the researchers actually compared this new mutation to others that have been found in dogs and even humans.
Oh, wow. And what did they find?
So interestingly, this exact complex mutation hasn't been reported in any other species.
Wow.
So it's really unique to this dog.
So this mixed breed dog is like a scientific discovery.
Absolutely.
Wow.
And this discovery could be really important.
Okay, how so?
Well, by knowing this specific mutation, researchers can now develop much more accurate genetic tests for hereditary myotonia in other dogs.
So this one dog could help a lot of other dogs.
Exactly. It could help breeders make better decisions.
Yeah.
And potentially even reduce how often this condition occurs.
That's amazing. Okay, so we've gone from this one dog with a weird gait to potentially helping a whole population of dogs.
It's pretty amazing.
That's really cool.
Yeah, and the knowledge from this case could also help research in humans, too.
Really?
Yeah, even though the exact mutation might be different, the underlying mechanisms and potential treatment targets
could be similar.
Could be very similar.
Wow, that's fascinating.
Yeah, it's a reminder that, you know, discoveries in veterinary medicine can often have implications for human health as well.
Yeah, One Health.
Exactly.
That's really cool.
Yeah.
So thinking about the future, could this discovery lead to new treatments for hereditary myotonia?
That's the burning question.
Yeah.
I mean, it's still early to say for sure.
Right.
But this discovery opens up some really exciting possibilities.
Like what?
Well, now that we know the exact genetic problem, we can start exploring things like gene therapy.
Oh, wow, gene therapy. So that would be like delivering a good copy of the gene?
Yeah, a functional copy of the CLCN1 gene.
To fix the problem.
Exactly, correcting the genetic defect.
So instead of just treating the symptoms,
right,
you're fixing the root cause.
Exactly. And of course, gene therapy is still pretty new.
Yeah.
And there are challenges.
Of course.
But this case gives us a really crucial piece of the puzzle.
Yeah.
And brings us one step closer to a potential cure.
That's amazing. I love that. So not only have we learned more about hereditary myotonia,
right,
but we're also closer to a treatment.
Oh.
That's really cool.
Yeah, it's a testament to the power of scientific inquiry.
Yeah.
And the dedication of all the researchers out there.
It all starts with one dog. That's amazing.
Yeah, this deep dive has been really enlightening.
It has. We've gone from a dog with a weird gait to a groundbreaking discovery.
It's amazing how one case can unravel so much.
And lead to so many new questions.
Yeah, really important questions for the future of vet.
It really shows that any patient, no matter their breed or background, can teach us something new.
Absolutely. And that even in unexpected places, there are incredible discoveries waiting to be made.
I love that.
This mixed breed dog, who started as just another patient, has become a catalyst for scientific progress.
What a powerful message.
Yeah, this deep dive has been a true testament to the power of observation, scientific curiosity, and the pursuit of knowledge in the veterinary field.
Couldn't have said it better myself. So as we wrap up this deep dive, what are like the main takeaways?
Well, I think the first one is that hereditary myotonia can occur in mixed breeds, even though it's often associated with purebred dogs.
Right.
And the second is that genetic testing is so important.
Yeah.
Not just for diagnosis, but also for understanding how these conditions are inherited.
Right, so we can prevent it.
Exactly.
With responsible breeding?
Through responsible breeding, yeah.
Okay. And I guess the last takeaway is that there's a lot of progress being made.
Oh, yeah, absolutely.
In veterinary genetics.
Yeah. There's so much potential for new treatments.
Like gene therapy?
Like gene therapy, exactly.
So that's really hopeful.
Yeah, definitely.
So, yeah, this case shows us that breakthroughs can happen in unexpected places.
Right.
Like with this mixed breed dog.
It all started with this one dog with an unusual gait.
And it highlights how important it is to be curious.
Yeah.
And to really pay attention to what we're seeing.
Right. And to keep asking questions.
Yeah. And to never stop learning.
Exactly.
Well, this has been a fantastic deep dive.
It has been fun.
I've learned so much.
Me too.
I hope our listeners have too.
From Vet Neurojar.
Keep those minds inspired.
Our hearts light.
And tails wagging.